Hi, everybody. It's Dr. Launderville. This is online podcast number 5. We're going to continue our walk through some signaling pathways. This one is called NFK beta. It's called the master transcriptional regulator of immune response. So it doesn't make antibodies as a result of this signaling, but it does make over 150 different genes get transcribed as a result of these transcription factors called NFK beta. And these include small antiviral peptides, anti-bacterial peptides. It stimulates the production of nitric oxide, which we talked about in the last podcast. And so very actively researched, especially now with COVID-19. So in the COVID-19 podcast, I talked about a cytokine storm that is happening in people that are having severe lung loss of function. So the people on ventilators, where there's just a huge amount of cytokines released over a short interval in a short amount of anatomical space. And this pathway would be behind a lot of that cytokine storm. So I've seen some papers where they're talking about specific drugs to inhibit this pathway. Okay, so how does the pathway work? This one, it sounds silly to say that it's complicated. They're all complicated, but this one particularly so. So the goal is to get these guys, P65 and P50, into the nucleus where they're going to be a transcription factor and they're going to bind to DNA, turn on those 150 so genes. Okay? So normally in the cytoplasm, those things are bound to an inhibitor called IK beta alpha, and these will stay in the cytoplasm. If they get phosphorylated, if IK beta alpha gets phosphorylated, then they will bind an E3 ligase, E3 ligase being our ubiquitin E3 ligase. E3 is the one that puts ubiquitins onto things, and then IK beta alpha gets polyubiquitinated and gets degraded. When it gets degraded, it goes away, and that releases or starts to show these nuclear localization signals that are on P65 and P50. Then they go to the nucleus, act as a transcription factor, bind to the DNA, turn on all those genes, transcribing all those genes that are part of the cytokine storm. So how do you get to the point where IK beta alpha gets phosphorylated? Well, there are kinases, IKK beta, IKK alpha, and together with a protein called NEMO that interact in this ubiquitin scaffold and get activated. This kinase gets activated and then eventually will phosphorylate IK beta alpha. So how does this complex, which includes a kinase, get activated? Here's one way it can get activated. So these are interleukin receptors that are in the cell membrane. They bind interleukin ligands, and then they recruit a bunch of cytoplasmic proteins as a result of that. One of those that they gets recruited is called TRAF6. TRAF6 is another kind of ubiquitin ligase. They're all over the place. Another kind of E3. It sets up sort of like this tent of polyubiquitination. So lysine 63, amino acid 63 on ubiquitin has another ubiquitin attached to it in this big chain. And this NEMO IKK beta IKK alpha complex binds to this tent. Once it binds to this tent, it recruits a kinase called TAC1, activates this kinase called TAC1. TAC1 phosphorylates IKK beta. That activates IKK beta, which is a kinase which will then activate IK beta alpha back on this page here. Okay? So we talked about how the goal was to get P65 and P50 into the nucleus. First, you got to get rid of this repressor. You get rid of that repressor by first phosphorylating it and then ubiquitinating it. What phosphorylates it? Well, this IKK beta has to be activated first. One way to get that activated is to polyubiquitinate interleukin receptors. Another way is to activate TNF alpha receptors. Another way is by ionizing radiation can activate this kinase, and virus infection can activate this kinase. And also toll-like receptors can activate these kinases. So all these sorts of things are environmental insults to cells. Something is going wrong. It's being attacked by a virus. It's receiving ionizing immunization. It's receiving some other part of your innate immune system signal, and that will eventually end up phosphorylating this kinase. That kinase phosphorylates other substrates, and eventually P65 and P50 go to the nucleus and stimulate transcription of all kinds of proteins that could help in the immune response, like inflammatory cytokines, chemokines, small antiviral and antibacterial proteins. As I said, this would be part of the cytokine storm. This signaling pathway would be part of the cytokine storm. And so drugs that may inhibit this pathway may be helpful in the current COVID-19 epidemic. So one thing about this to notice, that I would like you to notice, is that when we talked about Wnt and when we talked about hedgehog, ubiquitin tagging and subsequent proteolysis after getting ubiquitin tag, that would shut off Wnt signaling and shut off hedgehog signaling. In this case, for NFK beta, ubiquitin tagging actually turns on the signaling, because you have to have ubiquitin tagging of IK beta alpha, and first before that you have to have that ubiquitin tent set up, which I showed you on this through TRIF 6. Interacts with toll-like receptors. Toll-like receptors won the 2011 Nobel Prize. Another part of the inflammatory cytokine system, part of your immune system. And your book calls this, as I said in the beginning, a master transcriptional regulator of immune system in mammals. So over 150 different genes are activated by these transcription factors as a result of some kind of cell insult that may be associated with infection or ionization or receiving a signal from a different part of the immune system. Okay. Another pathway. I'd like to say it's the last one, but it's not. This one's called mTOR. It stands for mammalian target of rapamycin. So rapamycin is a antibiotic that was helpful in organ transplant patients, and this is first worked out in yeast, where they were figuring out this pathway, and they knew that the rapamycin was targeting sort of part of the pathway. They called that the target of rapamycin, or TOR. And then when they found the mammal version of all that, that's called mTOR. And there are mTORCs, which is mammalian target of rapamycin complex. And there's an mTORC1 and mTORC2. I'm mostly going to talk about mTORC1. So, this looks very complicated, and of course it is, but it's essentially what you have seen before in that when GTP is bound to this complex, it's on. And when GDP is bound to this complex, it's off. Okay? There are many things that will affect the ability of this to be in this state with GTP on versus in this state, GTP off. So independent of all this stuff, which I'll talk about in a second, it can just be nutrients affecting the ability of the equilibrium to shift toward more this way versus more that way. And if there are low nutrients available to the cell, you have a switch toward this side of the system. Okay? And that will lead to ribosome biogenesis, RNA polymerase III transcription, which is an RNA polymerase that is pretty sure involved in ribosome biosynthesis. Let me check that, make sure. And but Pol2 is the one for protein synthesis. But as far as protein synthesis go, there is a EIF4 binding protein. It's for EBP. In fact, there are several of them. And mTOR with its GTP state inhibits that binding protein because the binding protein is binding to EIF4 because it's inhibited from binding to EIF4 that releases EIF4. You remember that EIF4 is required at the initiation of translation to unwind any secondary structure in the RNA. It's got ATPase activity. And there's also S6K, which is a kinase that phosphorylates proteins on the small subunit of the ribosome and stimulates translation activity. So you release EIF4 and you stimulate translation activity by releasing EIF4 and also by phosphorylating some proteins in the small subunit of the ribosome. And it also inhibits autophagy. Autophagy is a phenomenon where vesicles are drawn or are built around large structures like mitochondria and ribosomes and then fused with lysosomes. It's a way for cells to die. Okay. You can also affect this equilibrium toward the on side or the off side by a activity of these two proteins, TSC2 and TSC1. They react to stress and hypoxia, and they will affect the equilibrium of toward more GTP bound to mTOR or more GDP bound to mTOR. The kinase AMPK can also affect the activity of this protein, which then can affect this equilibrium. And binding to a growth factor receptor, we've talked about other growth factor receptors like growth hormone, for example. The downstream effect of that can involve affecting the activity of this complex, which then affects this equilibrium. So, mTOR1, the one in this diagram, is responsive to growth environments. So environments where there are lots of nutrients around, lots of oxygen around, lots of growth factors around, mTOR1 is going to be active and stimulate translation. mTOR2 is not responsive to nutrients directly, but does get activated when insulin binds to the insulin receptor. So this is a pathway involved in sensing the nutrients that are in the cell environment and telling the cell, is this a good time to grow or not? Is there stuff out there to grow with or not? Okay. mTORC1, the mammalian target of rapamycin complex 1 responds to amino acids in lysosomes, oxygen, ATP, growth factor signaling. This all results in ribosome synthesis, increased rates of translation, cell growth. So these are things saying, yep, conditions are good, let's grow. And in terms of activating pathways that are going to kill cells, say let's not do that under these conditions, so inhibits autophagy. When cell stress is around, low ATP concentrations around, low nutrients around, low oxygen around, or rapamycin is around, like in a cell dish, then the activity of the equilibrium would switch toward more GDP bound and less of the mTOR complex being active. And mTORC2 responds to insulin signaling versus nutrients directly, but instead indirectly through insulin. The result of that is to reorganize this actin cytoskeleton. It's not sensitive to rapamycin, and it's inhibitory toward apoptosis. Again, we most of what you're going to run into in terms of a biochem class or this class would be mTORC1. Okay. Step back from just pathway-orama for a second to talk about development and cell fate. So, you started off as two cells fusing, and then those two cells eventually became a trillion cells that are you, and those cells are constantly replaced. How do cells decide that they're going to be a liver cell, a heart cell, a neuron, etc.? That's what I'm talking about when I'm talking about cell fate. So the original cell, when two haploid cells fuse and form one diploid cell, is called totipotent because it can literally become any kind of cell in the body. That's what happens. And how cells determine their fate is due to regulatory molecules. And these regulatory molecules usually form a gradient within the cell. They migrate to one pole of a cell, and that cell division, as a result of the concentration gradient of regulatory molecules, has a lot to do with how cells decide what they're going to do, what they're going to become. Programmed cell death or apoptosis is part of a developmental program, and most of the time you'll see that programmed cell death results from cells not receiving a signal. So in most of the pathways we've talked about how a signal arrives on the outside of the cell and then something happens as a result of that. When we talk about programmed cell death, we'll talk about that's the default, in that something is arriving on the outside of the cell, and as a result of that, the cell will not die. But if that stops happening, the thing stops arriving on the outside of the cells will die. And in early experiments on cell culture, that was something that was really hard to work out, and that what needed to be in the medium all the time in order for cells not to automatically go through cell death. Okay. When we talk about stem cells, we talk about harvesting cells that have multiple possible cell fates. Maybe not totipotent, they can become anything, but they can become lots of different things. So in early embryos, when they are just a few cells big, you can collect cells from a certain part of the embryo, culture them in cell culture, and then push them into different fates, become one sort of thing or another sort of thing. And a lot of this cell culture was documented and worked out in this fantastic book. I think I've mentioned it to before, The Immortal Life of Henrietta Lacks. She is the original donor of the original cells that could be kept in culture, so-called HeLa cells, named for her first name and last name. And I really encourage you to give it a read if you haven't read it. It's not a boring science book. Okay. So you have cells going through their first few divisions. There's not much growth phase, not much of a G1 phase, and not much time in between cell divisions. And so the cell is literally just dividing in half each time, and each cell, as a result of that, gets smaller. You'll answer that on H5P. Okay. There are things that you can do, experiments that you can do, that demonstrate that the signaling molecules are in a gradient and position of the cell matters. So let's say we have a 4-cell embryo, and we take one of those cells and we inject it into a embryo that is a little bit, few more cells in it, let's say 16 or 32 cell stage. Where the cells are implanted, what their position is, matters. So if you take your donor cell and you put it here, you get a different cell fate than if you put it here. Same cell, but if you just put it in different places, you get a different fate. So in terms of what the cell turns into. So there's something being made by these cells, and the proximity to that thing matters, so that there's a gradient of that signaling molecule that affects cell fate. All right. Some of the proteins that have been identified that affect how cells determine their cell fate are listed on this slide. One of them is DNMT1. It's a methyltransferase. It's a DNA methylation enzyme, and it maintains DNA methylation patterns. This is kept out of nucleus of very early embryos, so the cells of those nucleus exclude this enzyme, and those the DNA from those earlier embryos gets demethylated, so the pattern is removed, and then the methylation pattern resets as the tissue differentiates. So as the tissue turns into endoderm, ectoderm, mesoderm, they'll lay down a methylation pattern, and in fact that's essential for those cells to methylate. Other proteins that are involved in this process are OCT4, SOX2, and NANOG. These are the genes, these are the proteins that those genes transcribe. And these genes, or these gene products, these proteins, are important for whether or not the activity of these proteins is important, whether or not the cell is going to be pluripotent or totipotent. And they also repress differentiation pathways. So there are engineers, biological engineers, very much interested in reprogramming differentiated cells. So for example, if someone has a spinal cord injury, they want to deprogram those differentiated neurons back to an embryonic state when they could become anything, so they'll grow again. reactivating these proteins, which are usually only active in embryonic stem cells, is part of the effort to do those sorts of experiments. Okay. So you don't have to get stem cells from embryos. You can also get stem cells from adult differentiated cells. And you can get it from cells that can divide. Okay? So let's say you take an egg cell, remove its nucleus, replace that nucleus with a differentiated cell nucleus, like one from an olfactory neuron. That egg cell will divide and form an inner cell mass in a blastula. You harvest those cells. You import them into a blastocyst of a host cell, which will make the host cells will make a placenta, and the cells that you add from your embryonic stem cell will make the embryo, and then you can get a mouse. So this is not there. These cells are considered pluripotent, not totipotent, because you need the host cells to make the placenta. So the transplanted cells can't make the placenta. But these adult differentiated cells can act like stem cells, pluripotent but not totipotent, and can regenerate tissues and that sort of thing. And so there's lots of excitement about using this sort of technology to, say, make somebody a new liver or make somebody a new heart or something like that. The adult stem cells can also be made by deprogramming adult tissues. So there's a researcher called Yamanaka, and he uses protein mix of KLF4, SOX2, OCT4, and MYC. So some of these you saw here, SOX2 and OCT4. Okay, these are transcription factors that are important for activating genes for self-renewal and also repressing genes that allow differentiation. So you can take adult cells, let's say osteoblasts from a bone. You add this cocktail. It de-differentiates into just a stem cell, and then you add specific chemical gradients that can push the cell to a different cell fate. So you can turn these into fat tissue, for example, by adding a certain protein cocktail. And you can also go in this direction or in that direction. So a lot of these tissue engineering labs like to start with fat tissue because fat tissue is relatively easy to get from adults, and adults don't care so much to get rid of some of their fat tissue. So you can aspirate some fat tissue from someone, grow it in a dish, push it into another sort of cell fate, and then maybe make a patch, for example, for a heart that has been through a heart attack and part of the surface of the heart died during that heart attack. And so you take fat tissue from that patient, you grow some heart tissue in the lab, and then you implant a patch, and that person recovers. It also has all kinds of advantages because since you're using a person's own tissue, all their immune complement would be the same because it's their own tissue, and so you don't have the same sort of problems with tissue rejection than you would if you're getting tissue from somebody else. Okay, so with that very brief overview of cell fate, the big ideas being that there's a difference between totipotent and pluripotent, depending on how early the cells are harvested. The idea that you can harvest cells early in a cell embryo and push those cells to different cell fates depending on their position in a cell mass. The idea that there are proteins that are responsible for keeping cells in a less differentiated state, and you can use those proteins in a cocktail to take cells that are already differentiated, de-differentiate them, and push them into another fate. With all those ideas in your head, now I want to introduce the idea of programmed cell death and or apoptosis. So this is cell death as part of a developmental program on the way from going from one cell to a trillion cells. Some cells will die as a result of that on purpose. It has evolved that way. And an analogy I think is really useful for this is to think of building a skyscraper. So to build a skyscraper, you will see cranes on the skyscraper during construction. You'll see scaffolding on the outside of the building during construction so they can get the windows in from the outside, so they can lift up these huge iron beams from the street, place them in the building, and then once the building is done, all that stuff goes away. You take away the scaffolding, you take away the cranes. The same thing happens in you. So in order to build a brain, you have to have cells that are going to support that structure as it's developing, and then once the structure is there, they will be pruned, they will die through programmed cell death. And that's part of the developmental pathway. A lot of you are at the age right now where you're still going through that process in the development of your central nervous system. Programmed cell death is different from other kinds of cell death in that you see a very characteristic pattern of how the cell dies. The cell is divided into small vesicles called apoptotic bodies that are scooped up by phagocytic cells. Phagocytic cells like macrophages, white blood cells. And the DNA is cut at very specific places. So if you see if you extract DNA from an apoptotic cell, you get a ladder sort of pattern rather than a smear, which happens if a cell is dying by, say, necrosis. The cell got old, or the cell has been infected with something and caused it to die, then that DNA is degrading randomly, not specifically. Okay. A lot of apoptosis was figured out in our friends C. elegans. C. elegans have utili, which is a defined number of non-gonadal cells in adults. So there's 959 cells in an adult C. elegans. That, again, it doesn't include eggs or sperm. So an adult has 959 cells, and 131 of those cells will go through apoptosis during their developmental pathway. You can see which ones are going to go through apoptosis. They're all numbered. And we can say, okay, that cell is going to die within the next 3 hours, that sort of thing. And because of this huge advantage in this study organism, they could figure out what genes were active in those cells versus other cells. And then once those pathways were worked out, the mammalian counterpoints of those proteins were identified, and apoptosis was established as a really important pathway in cell development. Okay, another one for you on H5P. So, sticking with neurons and apoptosis. Here is 3 neural pathways, motor neurons here in blue, proprioceptive neurons that tell you where your body is in space in red, pain receptors, nociceptive neurons in light blue here, mechanoreceptors here in yellow, in wild type. If there are mutant mice that are missing neural growth factor or TRKA as a growth factor, then, for example, the nociceptive neurons go through programmed cell death and they disappear. If there are other mutants that are missing different growth factors, they go through programmed cell death and they are missing. So a big message that I want you to get out of apoptosis is the signal that arrives on the outside of the cell has to arrive all the time. You have to get your mail every day if you're a cell. And if you don't, the default pathway is you activate programmed cell death. Okay. So in nematodes, this was worked out. What are the steps and what are the genes that are activated, proteins that are translated during programmed cell death, and they were called CED for C. elegans death proteins. Once this was worked out, started to look for homologs in mammals, and this is what we have now, and many of these signals have been conserved, and this is the one we're going to focus on. Okay, back to our pathways. I know you were missing them. So we established that programmed cell death is a normal part of cell development. And how does programmed cell death work? So in the case of programmed cell death, you have to get a trophic factor constantly in order for the cell to stay alive. If the trophic factor does not arrive on the outside of the cell, then the default pathway is the cell will go through cell death. So this sort of thing we've seen before. These are something like a receptor tyrosine kinase, like a HER2 receptor. It receives its trophic factor, its ligand, it dimerizes, activates autophosphorylation on the kinase domain. That kinase gets activated, starts a kinase cascade, in this case through PI3 kinase, which phosphorylates protein kinase B, also known as AKT in other cells, and phosphorylates BAD. BAD is bound up by a protein called 1433, which you've seen before, and I'll remind you about that in a second. And then BAD can't interact with BCL2 or BCLXL depending on the cell. So BCL2 or BCLXL are inhibitors of these channel-forming proteins called BAC or BACs depending on the cell type. And these are in the outer mitochondrial membrane. If BAD is sequestered in the cell, that means that it cannot interact with BCL2 or BCLXL. BAD is an inhibitor of an inhibitor. So BCL2 or BCLXL is preventing this BAX or BAC from forming a channel. That's what it's doing normally. And BAD, when it's not sequestered, inhibits this inhibition, allowing the channel to form. Okay? So this is a little confusing. So this thing here in light green is an inhibitor, and it inhibits this thing here in blue from forming a channel. And that's what is normally happening in a healthy cell. And in a healthy cell, BAD would be phosphorylated and sequestered in the cytoplasm. When BAD is not phosphorylated because the trophic factor did not arrive, then it can interact with the inhibitor, and it's an inhibitor of the inhibitor. So it stops this light green molecule here, BCL2 or BCLXL, from inhibiting BACs or BAC, and therefore BAC or BACs can form a channel. Okay? So when BAD is allowed to interact with BCL2 or BCLXL. It stops it from holding back or backs, and then back or backs will form a channel. When that channel is formed in the outer mitochondrial membrane, cytochrome C, which is normally part of the electron transport chain, leaves the mitochondrion and enters the cytoplasm. This is unusual. Cytochrome C is not normally a cytoplasmic protein, and one of the hallmarks of apoptosis to find out is this cell going through apoptosis is cytochrome C in the cytoplasm. So this is often a bone of contention among reviewers. Let's say you're studying something that you think kills cells, and the reviewers are going to want to know, were those cells killed because they just died from necrosis, or were they killed by apoptosis? And to show that it is or is not apoptosis, you can show if cytochrome C is in the cytoplasm, then it's apoptosis. There are other ways too. Okay, so cytochrome C gets released from the mitochondrion, is in the cytoplasm. It binds to this complex called APAF1. APAF1 activates a pro-caspase. So pro-caspase 9 is a protease. It's a protease wannabe, in that the pro form of the enzyme is not the active form of the enzyme. The active form is caspase 9. So APAF1 turns pro-caspase 9, which is inactive, into caspase 9, which is active. And caspase stands for cysteine-dependent aspartate-directed protease. So these are proteases. They cut proteins. And this protein then cuts this protein, turning it from a pro-caspase into a caspase. And now there's a caspase cascade, like you saw before, a kinase cascade. One protease activates another protease, activates another protease, and then all of those proteases cut up things inside the cell, cut up the DNA, cut up proteins in the cell, and the cell dies. Okay, so when BAD is phosphorylated, sequestered in the cytoplasm by 1433, if that doesn't happen because the trophic factor didn't arrive, then it inhibits this inhibitor, allowing the channel to form. What are other ways that apoptosis can get activated? Well, it could be that you get ripped off your basement membrane because there's mechanical damage in the cell. Okay? If that's the case, then there's a protein called BIM that acts like BAD. It is an inhibitor of an inhibitor. So it's going to inhibit BCL2 or BCLXL, depending on the cell type, and then allow back or backs to form a channel. What happens if there's been massive DNA damage? Let's say you're a cell that is differentiated and it's hanging out in G0 for years. And during that time, there's been some DNA damage. Now you're going through a cell checkpoint. The cell checkpoint is asking, has there been DNA damage? And the answer is yes. The first thing the cell is going to try to do is recruit DNA repair mechanisms, which you learned about in cell and molecular biology. If they fail, however, PUMA will be released from the nucleus, and PUMA acts just like BAD in that it inhibits inhibitors. It also can promote formation of the channel, and cytochrome C is released and apoptosis happens. Okay, so these are all situations in which you want the cell to die. There's something wrong. Trophic factor is not arriving. The cell is mechanically damaged. There's been DNA damage to that cell, and you don't want to replicate that cell, and so you want to go through programmed cell death. We saw 1433 before in the kinase cascade, where RAS activates RAF, activates MEK, activates MAP kinase, and it does the same sort of thing in that it binds up phosphorylated residues on proteins and keeps it in an inactive state. This will be on your H5P question. Okay, APAF1, this thing back here, is actually a giant complex in cells. And in worms, this is called the hepatosome. In mammals, it's called APAF1. It has the same sort of structure. And in your book, they call it a 1.4 megadalton wheel of death, which is a fantastic name. I think this is like an album cover from a speed metal band or something like that. So it has a compartment in it where the caspase go from procaspase to caspase, and then you start this caspase cascade. And again, this is known, the hepatosome is known as APAF1 in mammals, and the hepatosome in C. elegans. Other cell types use apoptosis pathways to kill cells. Your immune system has cells that kill other cells as part of their function. Macrophages can kill cells through lots of different mechanisms. Natural killer cells can kill cells. One way that both macrophages and natural killer cells can kill cells are through death signals. In macrophages, it's in a molecule called TNF-alpha, which we'll talk a little bit more about. And in natural killer cells, it's called FAS. This will be presented on one cell interacting with the cell that's going to die in a thing called a death receptor. And that death receptor will activate the familiar apoptosis pathway. This is the sort of thing that's happening in COVID-19 patients where cells are being activated. The apoptosis pathway is being activated in cells, and this TNF-alpha molecule, this death signal, is maybe over expressed in COVID-19 patients. There's a paper from last week where they looked at various cytokines expressed in severe cases versus moderate cases of COVID-19. Severe cases would be people who are intubated on a respirator, and they looked at concentrations of TNF-alpha in people with severe cases versus moderate cases, and is significantly higher. I'm going to ask you to do an exercise at home called a concept map. This is something I like to use a lot on tests. Hint, hint. The reason I like to use it is a great way if you understand how things are related to each other. So how they work is like this. These things in the little ovals are concepts. They're ideas that we've talked about in class. We've talked about cyclic GMP, we've talked about STAT, we've talked about GPCR, we've talked about HER2. And I want you to make connections among these concepts and connections that are substantive. So for example, I might draw an arrow between cyclic GMP and GPCR, and I would write over that arrow, cyclic GMP is a second messenger produced by guanylyl cyclase after activation by a GPCR. So I, reading that as a professor, would say, okay, this person understands a connection between these two in a non-trivial way. So a non-trivial way would be like trivial way would be like cyclic GMP and GPCR both have the letter G in them. Here's another example. STAT, connection to HER2, is activated by phosphorylation of JAK receptor, which is a separate polypeptide, unlike HER2, which is a type of RTK receptor in which receptor and kinase are all one polypeptide. So that gives me a lot of knowledge about how you understand those two things are connected. Something like GPCR is a protein, no good. It's no good because it's just too trivial and too general, and it's not making a connection between two concepts. It's just listing out a fact about one concept, and so that's no good. That would not be acceptable. Okay, so for homework in this week, There is a concept map on Brightspace. I want you to print it off or mark it up on your screen if you've got a tablet, or you can reproduce those concepts in a Word file and then make your own connections. Somehow make this concept map with the example that I've got on there. Okay, so you can, if you don't have a printer at home, you can just make a Word file with these concepts and then make connections among them. Okay, you can mark up a picture, so you can take a picture of that thing and then mark it up and then submit that. You can, if you do have a printer, you can print it off and then draw it by hand. Make sure that I can read it. It's got to be legible, otherwise I'm not going to be able to grade it. And then that's due in a week, and it'll be worth as much as a quiz, so 10 points. Okay? Anybody is having trouble getting that done because you're having technology issues or whatever, please just contact me.